Thank you so much, Manu, and really the huge team behind, beside, and around you, including Theo Michele for organizing all of Ocean Visions. And this talk series has been a real shot in the arm and exciting for me. So I'm really delighted to do this and to move forward. So my starting point, as probably everybody here, is always our global ocean and the reminder that we're an ocean planet, and any new solutions we develop that are ocean-based will have huge consequences on our planet. Today, I'm talking to you from the Salish Sea, and I don't know if you can see my cursor here, but my little island is right here. I'm actually right at that spot on this little bay. So this is Vancouver Island. This is an amazing inland sea that's transboundary, encompasses the US and Canada. Here's another view of it. And I'll come back to some of the Salish Sea issues as I go along. But I do want to start with a reminder that natural systems in the Puget Sound Basin alone, so that would just be this, not the whole Salish Sea, if valued, would be worth between 300 billion and 2.6 trillion, and probably more, because a lot of these valuations are underestimates, as I'll get to. So I think I'm kind of preaching to the choir here when I say that natural capital investments often present a low financial risk while providing a high level of benefits. And I thought about this a bit. A productive life is often significantly longer than that of built capital investments. And I'm just showing a few of the highlights of groups that are working on this, including to remind you about Nancy Knowlton's fantastic talk in this series, which highlighted a few of those contributions for natural capital. So I'm going to talk in part about some projects that are based in the Salish Sea and a little bit of a pitch about natural capital here. And I'm choosing it kind of as a demonstration project. It's a transboundary sea. And so picking these kinds of projects kind of take us right head to head with one of the issues we have in the ocean, of course, which is our international boundaries that the oceans don't respect. We have unusually high value marine resources. Of course, everybody knows about our salmon, crab, oysters. Probably far exceeding the value of these are some of our nature services. And again, a reason to highlight this region is the high value of some of the nature services, the intactness of some of these ecosystems. And I'll talk about seagrass meadows. So I think anything we do here as a demonstration or getting the ball rolling will produce a high return on investment. And that's a good thing, because right now we are in an extremely tough spot with a very high threat of decline with future warming. And so what I want to talk about today are nature's superpowers, as I'm calling them, for a healthier ocean. And I'll start with the bad news, which is infectious epidemics in the ocean increase with warming. And unfortunately, this is the work I do, so I kind of confront this every day. But at the same time, we're looking ahead to solutions that involve some of our natural capital or natural ecosystems. And I'll talk about examples we have. I'm not even going to talk about the new frontiers, but when I get thinking about it, I get so excited because we have not touched the potential of this. We have not touched the potential of marine microbiomes, phage therapy, or oil-eating microbes to help us improve the health of our oceans. And as I noted, Vera Trainer's here, and one of the bacteria that lives on the surface of eelgrass blades actually impacts the growth of harmful algal blooms. And so this is of great interest to her research group. And there's lots more that do other things we haven't even identified. This little bottle here, I just pulled off Amazon. These are ultra microbes that eat oil. So if you have an oil spill, you can pay $5 and get some of these and sprinkle them on your spill. And so I want to then move forward at the end to talk about a way forward with natural capital. This is what we do. We study the ecology of host-pathogen interactions. And I think of these as sentinels of change in our balance of nature. And so we're always studying host-pathogen interactions, but as ecologists, it's always in the context of the environment, which, as everybody here on this call knows, is changing rapidly and not in a good way. It's changing in a way that favors these outbreaks by getting warmer. And so today I'll just talk about our eelgrass and sea star work, because this kind of spans ecological interactions from being a foundation species that's an ecological engineer to a keystone that actually creates some of the structure in our marine habitats and weighs in on our balance. This is just a little bit of background for anybody that hasn't really thought much about this. A lot of the infectious diseases of marine taxa are temperature sensitive. And so when oceans warm, these things outbreak or get worse. And these are the ones, these are some of the big ones. Everybody knows kind of all these examples of cases where we've had outbreaks. And I'm just kind of showing that to give you a little background. I'm going to highlight just two of them today. I want to talk about our sea star outbreaks from wasting disease. And this is kind of what happens to one of those species, the sunflower star. When it picks up this agent, its arms curl and then sadly and in a very gory way fall off, and then it dies. It's a big deal because this started in 2013, and it is the largest marine wildlife epizootic we know of in terms of the numbers of species, which are over 20, and the range, our entire West Coast from Mexico to Alaska, but also over into Asia. And so a very wide span event. It turns out that it's affected ecologically important species. We've just listed the sunflower star with IUCN as an endangered species. This was as common as a robin in our subtidal waters, no kidding, before this happened. And so it's been a big change. And there's a warming component. I'm going to briefly share this video because it's a video abstract of a paper we published that kind of gives a better picture of this than my telling you. I won't do the whole thing, but let me just start it.
There's an epidemic in the ocean. Since 2013, a viral disease has been turning sea stars in the Northeast Pacific into melted piles of goo. Of the 20 or so species of sea stars affected by the virus, one of the hardest hit were sunflower stars. Until now, we haven't known just how bad the decline was, but new research has begun to reveal the longer-term continental scale impact of the epidemic on certain species. Scientists in the US are now suggesting we formally list the once common sunflower star as an endangered species. Trained citizen science divers from California to Alaska counted sunflower stars on over 11,000 dives, while scientific divers from the Hackeye Institute carried out more detailed surveys on the BC Central Coast. When they looked at all the data, scientists noticed something in common where they saw outbreaks of the virus: anomalously warm water. We still don't know how these warm water anomalies and the virus interact, but researchers say these warmer than normal water temperatures were related to dramatic sea star declines. While divers can patrol waters near the surface, we didn't know whether sunflower sea stars might have found refuge at deeper depths. But thousands of NOAA bottom trawl surveys have revealed that when it comes to sunflower stars, the disease didn't stay in the shallows. For example, data from Washington State shows a crash in populations in both shallow nearshore and deep offshore environments after the epidemic began in 2013. Data from other areas on the coast are similar, with population declines of as much as 80 to 100% in areas across the 3,000 kilometers from Alaska to California.
So I'm going to stop that now. I just wanted to make the point that these kinds of, although there are more questions than answers in this one, these kinds of wide-scale events are likely to increase with warming. This one is having cascading impacts ecologically because of the importance of many of these stars as predators, and at least in the case of the sunflower star, which is remaining to this day at 0 in California. One of its prey items has exploded and is knocking back kelp beds. And so while the causes of kelp bed decline are likely complicated, this is definitely a big player in that. And so it's an example of kind of this ecological cascade of a bad event. I want to move to eelgrass meadows because this is a vital marine habitat with important services. And again, I know many of you know all about these. Particularly, there's been a lot of interest in blue carbon and climate mitigation role of ecosystems like seagrasses. What I want to do is contribute filtration services and emphasize the value of that and the potential for seagrass to reduce disease risk for humans in corals, and we don't even know other species. However, the plot thickens because this habitat itself is affected by an infectious agent. And as opposed to the sea star one, we know a lot about this and have good studies on the causative agent. These lesions are caused by a protozoan, Labyrinthiella zoster, and I'll tell you just a little bit about our work on the West Coast. And just, this is what it looks like inside the eelgrass tissue, and then it's really alive. So there you go. This is just a video showing those spindle-shaped cells running through their mucus networks inside their hosts as they're eating away the tissue. This is a project funded by the National Science Foundation, led by Emmett Duffy, with co-PIs Carla Gomes, Tim Hawthorne, and Jay Stackowitz, and a ton of really key West Coast partners. And our goal was to assess along the entire West Coast the prevalence of this disease and the level and the potential for it to do damage and potential climate links. And it brought in some new technologies with drone surveillance, as well as artificial intelligence to actually do the diagnostic identification work on our eelgrass surveys. And I wish I could tell you about the AI stuff because it's cool. And in fact, it even won a prize at a very prominent AI conference, but I just don't have time today. But the results of the survey, and these are thousands, several thousand blades surveyed: Alaska, BC, Washington, Oregon, California, Bodega, and San Diego across this cline. And what you can see is that almost 75% of the blades in our sites, which we think of as pretty good places, have this disease. Similarly, there's high levels in Bodega and in Alaska. The other part of our study, and we did just submit a paper this week, so we're moving along with it, is to show climate temperature links. And so in this case, these are our, I better slow down here. These are our sites. So this is Oregon, this is Washington, this is Alaska. This is the prevalence of the disease. So what percentage of the blades that we sample actually have the disease? And we've been studying this for over a decade, so we have good diagnostics on this now. And then how does it plot against temperature anomaly, not absolute temperature, but the anomalous, well, in this case, above 0 warmings. What you can see is that the higher prevalence situations for 2019, this is our first year, were all places where there's a positive temperature anomaly: Washington State and Alaska. Places that were cold in that year, and the Oregon site was unusually cold, 1.5 degree negative, showed a very low level of disease. And so, you know, this is early days, first year of our project, but we're really, really sort of, well, pleased to have the data and to present this. So we have a major habitat like that could be part of one of our solutions. It's also affected by disease. Now I want to kind of turn to the issues of disease as a sort of a larger issue that affects humans, not just starfish, salmon, eelgrass and other things, and also some of the problems we have with introducing pathogens into the water or pathogen pollution. So this is a study that led by Jolie Lamb and Rachel Littman, where they mapped out how coastal urbanization in Myanmar influenced human pathogens in food, such as mussels in normal beds. And then here's a news report from Seattle, King County fined for sewer overflow violations. So this is getting into our food and causing problems. So I did just release a book a year or so ago, Ocean Outbreak, to kind of bring all these stories together. And I'm going to just mention it very briefly here, because I talk again about this theme of infectious disease outbreaks being fueled by warming and increasing, and sort of being an increasing problem for us. But really, one of the reasons to write this is to talk about natural marine ecosystems and some of our solutions. And so I'm going to move to that. This is a paper that we published in 2017. It was part of a ecosystem services project in Indonesia and the Philippines, run by the Global Environmental Fund, to figure out ecosystem services and how they could be kind of wed into marine spatial planning. And this was really why we kind of did this project, because we had noticed that the seagrasses were adjacent to healthy corals. And we thought, well, gee, we wonder if we could measure an ecosystem service in our seagrass beds in Indonesia. The result is this paper, led by then postdoc Jolie Lamb, that pathogenic bacteria were reduced by 50% in these seagrass beds replicated across 4 islands. And so I'm going to just show you again, this is kind of an old paper, right? It's a few years old, just a little bit of it. This is that island. This is Barring Lompo. You can see what these islands look like. These are beautiful marine habitats, but with enormous sewage pressure from places with untreated sewage. And it's in Sulawesi here. So the reason we started this study is we all got sick there. So I feel like I can speak to the issue of human health. The most clear take home here is if you look at these Enterococcus indicator bacteria, in the places where seagrasses were absent, you can see lots of little red dots on our culture plates, thousands of them. In the places where seagrasses were present, you don't see very many. So this kind of led us to doing these transects from the shore to the seagrass beds to the reef, inside and outside places where there were seagrass beds. And so you can see for our indicator bacterium, the Enterococcus, that there is in fact a 50-fold reduction from levels that are the same at shore in the seagrass. And so this motivated Jolie to go back and sequence all of the bacteria, including identifying quite a few human pathogens that were also reduced in these seagrass beds. We wanted to kind of go full circle and make the link between potential for human health and potential for environmental health. And so Jolie surveyed the corals, and you can see that their levels of coral disease, it's taking over. The levels of coral disease were roughly half or less inside the seagrass beds relative to just outside for 2 of these major coral diseases. So this kind of sets up the possibility of a one health situation where both humans and biota could benefit from clean living and having natural ecosystems. I want to go back now to the Salish Sea because we wanted to replicate some of this work in a more urban setting and in a setting that was in our country to show the generality of it. So this is Jolie here and PhD student Phoebe Dawkins out setting these cages in Puget Sound with mussels in them. What we did is we used the mussels to be passive integrators of the bacteria. And so The study was a combination experimental work where these mussels, we put them out inside and outside seagrass beds, and then some that we took advantage of from the Mussel Watch program. And this work is about to be submitted, but it's still in prep. And what it showed is that the relative abundance of potential human pathogens was significantly higher where seagrass was absent relative to where it was present. So this is again in Puget Sound. And the DNA sequencing work showed potential human bacterial pathogens belonging to about 33 species. And I don't have time to talk about what all of them were, but the result is that in these urbanized areas, seagrass meadows showed lower levels of human pathogens in the mussel gills. Just a point about how bad these things are, they're bad. Now, they're not always in the water, so I don't want to be alarmist about this. These are sort of intermittent, which was why the mussels were a good idea, because they're sort of constantly taking in these bacteria over time. But it's things like Campylobacteria, Haemophilus parainfluenzae, Enterococcus faecalis, and Staphylococcus hemolyticus are just 4 of the 33 species identified. So they're bad. I don't have time. I'm just going to flash this by you because I want to get to the discussion. But of course, it's probably not just seagrasses. Other natural ecosystems have, and I'm calling them superpowers, and I don't mean that to sound flippant, but I guess I want to allude to the fact that there's processes we don't understand yet that are new and novel. So coral disease is also lower in marine protected areas. And then here's a couple of the studies, one by Lori Raimondo in 2009, one by Kathy Page just about the same time. And then some more recent paper by Jolie Lamb in 2016 from her PhD thesis, showing, again, overall disease prevalence lower in all these areas with marine protected areas versus those where fishing occurs. So just saying. So I'm going to kind of wrap up here by saying that I've talked about the danger of infectious epidemics increasing with warming. Of course, we know there's other things that are increasing with warming, but it's particularly pernicious in that these infectious diseases synergize with other factors such as pollution, warming, and other issues in the ocean. And so they can be particularly problematic and change unpredictably. We've shown seagrasses have superpowers for ocean cleansing. There's other opportunities for cleaning up the ocean using our natural biota. So how do we reprioritize and scale up solutions? I want to give a shout out to the Natural Capital Project, which is based at Stanford, and the Nature Conservancy, for making science accessible and actionable to drive values of nature into decision making. And we're going to be looking to them for help with this. They already have many implementing partners worldwide. So the question for our project is taking our science discovery that seagrass and other wetlands impact human and biodiversity health and transform that into programs that conserve and restore these habitats. So maybe you're thinking, well, gee, we already know these habitats are important. We already know they're really great. But I think health is particularly important and has enormous economic value. And so our expectation is that if we're actually adding the health value in, it will make a difference. And so I just want to make the point here that, for example, in the Nature Conservancy resilience calculators, they weigh heavily on valuing, for example, eelgrass for its climate mitigation and blue carbon values, its habitat and pollution control of nutrients and toxins, but there's nothing for health at this point. And so what we're talking about is adding new co-benefits of human health that we think are substantial. And so the other part of this is, okay, so our job is to go and realistically value eelgrass. We'll get on that. once we do that, that should incentivize more preservation of eelgrass and some of these other habitats. I think what we're going to find is the cost of preservation is relatively low and the benefits are immediate by identifying low-hanging possibilities for preserving some of these habitats. Along the way, restoration becomes important, but this is very valuable, but it can be costly and slow. In a region such as the Salish Sea, where producing an annual report card, I think, would show a very large return on investment. And so that's kind of what I'm suggesting, is that with some of these regional projects, we really look for places where we can make a strong demonstration on it. And I'm not sure how long I've taken, but I'm thinking it's about 30 minutes and I should stop right here and kick off discussion of this topic.
You still have some minutes, but we're happy to start the discussion. Thank you. Thank you so much.